So we started to gather more evidence, and one of the first things we had is a guy named Char Gaff came through and started analyzing the amounts of the various types of nucleotides. So there's 4 basic types of nucleotides in DNA. They all have essentially the same sugar, deoxyribose. They all have a phosphate group. What they do is they vary in terms of which nitrogen base they have. So they can have any of these 4. So he started analyzing in different species how much adenine they had, how much thymine they had, how much guanine they had, and how much cytosine. And what he found was there was a relationship, that species would have roughly the same amount of adenine and thymine and roughly the same amount of guanine and cytosine. So A equals T, C equals G. So this is kind of cool. It gave us some evidence that there's some relationship going on here between adenine and thymine, guanine and cytosine, but it also allowed us to do some basic math. So if I tell you you have 15% cytosine, you should realize that I have to have 15% guanine, because they have to be equal. Well, if I total that up, that's 30%, which means I still have 70% left over. And because that has to be split evenly between adenine and thymine, that means that half of 70%, or 35%, must be adenine, and that means 35% must be thymine, because it has to be the same amount. So altogether, we have 100%. But this is kind of a cool way where we could break things down if we know even the amount of a single type of nucleotide. So some evidence as well as some ability for us to do a little bit of math and figure out stuff about a species. That was Char Gaff. Then you had Rosalind Franklin. She figured out how to use X-ray diffraction, which is a way of taking like an X-ray image of DNA. Now when you look at it, it's not like this is going to look where you go, oh yeah, that's DNA, it's got to be this shape. But this was critical because by having this X-ray diffraction pattern, you could build models. You could kind of test out what the structure of DNA might be, and then you could do the calculations to figure out, would that give me an image like they found? So I can use Rosalind Franklin's X-ray diffraction image as a way of checking my answers. It doesn't give me the right answer, but it lets me see if I have the right answer. And so Watson and Crick were 2 of the guys that actually discovered the double helix, the actual structure of DNA, and they did so by using Rosalind Franklin's image so they could keep building models and checking their work. So it's kind of trial and error where they'd say, all right, let's build this and see if it's got 3 strands. What would that do? Nope, that doesn't fit the image. It wouldn't give us the proper picture, so therefore it can't be right. So they just kept rebuilding it, and they stick the nitrogen bases inside, outside, up and down. They just kept rearranging stuff until they got something that fit this particular X-ray diffraction pattern, and that happened to be the double helix, which we now know as the structure of DNA. So the double helix is commonly also called the twisted ladder, because it's a lot like you take a traditional ladder and you just started twisting it. The sides of the ladder we tend to call the backbone, and that's made up of phosphates attached to sugar, attached to a phosphate, attached to a sugar, etc. Now, in the middle, the rungs are going to be the nitrogen bases. So they're going to stick out together, and then they are going to connect to one another with hydrogen bonds. So essentially, this middle part is where it's weakest, because these are hydrogen bonds, whereas all the other bonds, every other bond in the structure is going to be covalent. So it's going to be strong. It's going to be very, very powerful. But it's weakest right in the middle here, where the halves of the rungs meet. Those are hydrogen bonds. Those are not as strong. So if you're going to have DNA separate, it typically unzips. It typically separates down the center. Now they figured out the way that Char Gaff's rules tie into this is because adenine can only properly hydrogen bond to thymine, whereas cytosine can only properly hydrogen bond to guanine. This works because each of them have different sites where they can hydrogen bond. So adenine and thymine have 2 sites where they can do this, and it's kind of in a certain position, whereas cytosine and guanine have 3 sites that this happens, and they're in a different position. So if you try to line like a cytosine and an adenine or a cytosine and a thymine up, they wouldn't properly align, so they would not be able to stick to each other. And so this helps ensure that we've got this complementary base pairing system is what they call this, where cytosine's always going to bind to guanine, adenine's always going to bind to thymine. This is very important because it makes it pretty easy to replicate or copy DNA. Because even if you've got just 1/2 of DNA, you know that if there's a cytosine that's exposed, that doesn't have a partner, so we've just got like half the ladder, partial rungs, you know what other partial rung wants to come in there. If it's a cytosine exposed, only guanine wants to, or in many cases can, come in there and bind to it. Otherwise, it wouldn't function, they wouldn't stick properly, so it wouldn't work. So this makes DNA very reliable, allows DNA to make copies of itself very, very consistently, so we don't get so many mutations that we have people dying left and right all the time from mutations, which would not be good for the survival of a species. I mean, it's important that your genetic code's pretty consistent. Some mutations can be good, but too many mutations in general tends to be bad, because most mutations, if they have an effect, it's a bad effect. So you don't want to have a ton of mutations. That's a recipe for disaster. So looking at this, you can see we've got phosphate, sugar, phosphate, sugar, phosphate, sugar. That's the backbone that we talked about. You can see the nitrogen bases here, and they're coded accordingly. So you can see there's different colors for adenine, but you can see adenine always binds to thymine. This also makes sense because you'll notice that one of them that has 2 rings, the guys with 2 rings are called purines, will always bind to a guy that only has 1 ring. Those guys are called pyrimidines. I'm not going to make you memorize this much, but keep in mind you need to have where a 2 binds to a 1 because it keeps the spacing proper. If you tried to have something like an adenine bind to a guanine, another guy that has 2 rings, they'd actually be spaced too far apart. They wouldn't be able to lock in together. It would make the gap improper. Same thing as if you try to have a thymine bind to a cytosine, there'd be too much space there, so they wouldn't properly connect because this backbone is a set distance apart. And so there's always going to be where adenine binds to thymine, guanine binds to cytosine, because it also keeps the spacing proper. And so this occurs down the way. You can see the 2 hydrogen bonds versus when you have guanine and cytosine, you can see the 3 hydrogen bonds. And as we said, this is the weak part that's down the center, whereas on the sides here, this is going to be that strong backbone that we talk about. Now, DNA doesn't exist just by itself. How this is going to work is DNA is going to be wrapped around a series of proteins. They happen to be called histones, but they're going to wrap around it. And then you get these long chains then called nucleosomes that are just a bunch of DNA wrapped around a bunch of histones. So this nucleosome idea is pretty much what you think of when you think of chromatin, when DNA is loose, when DNA is accessible. When we want to do something like cell division and we want to pack the DNA so we can move it easily, we can then pack together these histone proteins. We can then take these packed together histone proteins that make this thick fiber, and we can start to coil it back and forth to make an even thicker fiber. We can then take those thicker fibers and we can go ahead and coil them back and forth to get even thicker fibers, and we keep doing that until we get this chromosome. And so the chromosome is essentially just going to be highly, highly, highly compacted DNA to form these thicker fibers, and then we coil those thicker fibers to get even thicker fibers, and we coil those to get even thicker fibers. So really, 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 when you look at your chromosomes, it's just a whole bunch of very, very tightly coiled DNA. At this point, we don't keep chromosomes much because they're so tightly coiled that the DNA is inaccessible, so we can't use the DNA. This is why we only form chromosomes when we're about to move the actual DNA around for something like cell division, and then immediately thereafter we go back to chromatin, because when you're looking at chromatin, the DNA is accessible so things can come in and access it. They can attach to it. We can use it.